A cyclic sugar has a hemiacetal at the anomeric carbon. That carbon has two oxygens attached to it, the ring oxygen and the hydroxyl group. That hydroxyl group is easily replaced by an alcohol to make an acetal. When that alcohol is a sugar, you make a disaccharide. The oxygen of this sugar hydroxyl group is the oxygen that ties these two sugar units together. We've got two units hooked together, so we call it a disaccharide. But notice that the disaccharide has an anomeric center that is a hemiacetal. So this hydroxyl group can be replaced to make a trisaccharide. And that third sugar unit can have an anomeric center that lets you hook another one to it, and so on and so forth. Chains of saccharide units can be built up this way. I'm showing you here 3 alpha-D-glucose units hooked together. This saccharide chain could extend past where this wavy line is. But 3 units let us see all we need to know. It turns out that the stereochemistry at each anomeric center is really important. These are all alpha because the oxygen is ticking down. So we call it polyalpha-D-glucose. Now these Hayworth representations let us see some things, but they don't let us see the way the molecule really looks in space. Here's a drawing that shows these sugar units in the chair form. Again, we can see the anomeric stereochemistry, and this chain can extend for literally thousands of units. That's a polysaccharide. Now Mother Nature has this all worked out, and polysaccharides that are essentially straight chain alpha-D-glucose are known. It's called amylose. Now when you look at this, it's easy to see that the other hydroxyl groups here could be used to hook other sugars to them, and Mother Nature has that worked out too. Take a look. I've written the same trisaccharide here as the start of a polysaccharide, but now on the second sugar unit I'm showing another sugar unit hooked to it. This leads to branching, and of course a hydroxyl group like this could be used to hook another sugar in this direction. So this branching could be extensive. There are 2 important types of polysaccharides that have branching like this. One is called Amylopectin. The other is called glycogen. It turns out for glycogen, about 10% of the linear sugar units have branching, while about 3% of the amylopectin sugar units have branching. The glycogen, which our body uses as the first store of energy, is the most branched of the carbohydrates. Both amylose and amylopectin occur in starch. And starch is 20 to 25% amylose and 75 to 80% amylopectin. So it's interesting. We have these linear polysaccharides. If they have the alpha stereochemistry, we call it amylose, amylopectin, or glycogen, and they can have significant branching. It's the fact that it has alpha stereochemistry at the anomeric center that lets our bodies metabolize starch to get energy. If we have the beta stereochemistry for the polysaccharide, we can't metabolize that polysaccharide. Here's a drawing of Hayworth structures linked together as a polysaccharide that has the beta anomeric stereochemistry. And you can tell just by looking at the way these structures are drawn that this polysaccharide has something different about its structure. In fact, it's quite different. But let's take a look at the chair forms first to take a better look at exactly what it looks like. Here it's represented in the chair form, and you can see, it doesn't look too different from the alpha stereochemistry. Except, for instance, this middle sugar would be sticking down rather than straight across. And that turns out to make a huge difference. Because these sugar units are hooked together by single bonds, it's possible to see rotation of one relative to the other. For example, we can have rotation about this bond, and simultaneously this bond, which rotates the whole molecule here. When it rotates 180 degrees, it's turned all the way around, and this oxygen is on the other side. Now here's the cyclic oxygen sticking out toward us, which is not the way we usually draw it, but it's the way it sits in nature, because look what happens. When that sugar unit is rotated relative to the other 2 next to it, all this extensive hydrogen bonding that I show you here occurs to form a rigid structure, we have 4 new intermolecular hydrogen bonds that keeps this structure from bending. This poly-beta-D glucose is called cellulose, and it's the rigid sugar that gives wood its structure. Think of it. With the alpha stereochemistry, the poly-D glucose is very soft, like starch. But if you just change the anomeric stereochemistry to beta, the polymer can establish a whole bunch of intermolecular hydrogen bonds that holds it perfectly rigid. Think of a baseball bat, for instance. Now, if you wonder whether hydrogen bonding could really do something like this, just think about water. When water freezes to make ice, that ice structure is held together simply by hydrogen bonds. And look at how hard ice is. It's not something that you want to try to break with your fist. One other interesting detail about the stereochemistry of poly-D glucose. We can metabolize the alpha stereochemistry, so amylose and amylopectin are polymers that we can digest. We have enzymes that break these down into the individual carbohydrate units, which we then use for sugar. In contrast, we do not have enzymes that will break the beta anomeric polysugars. So we can't get any energy from cellulose. And yet we feed hay to horses. Horses, like other animals, can't metabolize the beta stereochemistry either. And so cellulose is of no use to them for energy until the microbes in their gut, which can metabolize the beta stereochemistry, break it down into sugar units. So horses, like other animals that eat hay, depend on microbes in their gut to do the initial stages of metabolism to turn cellulose into sugars. The take-home: the stereochemistry at the anomeric center of poly-D glucose is terribly important.